mapping generates a BUM or a SUM file. So no longer FASTQ files, but BUM and SUM files. So you have seen that these BUM and SUM files, they contain a lot of information. So for every read that could be mapped, typically, they contain all the information from the FASTQ file, so the read itself, the sequence, but also the identifier and also the quality scores. And these BUM and SUM files, they also contain, of course, the information of the mapping. So this part is the information on the mapping. So that means that these BUM and SUM files, they are very big. Yeah? You have all the things from the FASTQ file and additionally the information of the mapping. That's a lot of information. So, and that makes it again very hard to access them and to search through them in a fast and efficient way. Just like we have seen for the reference genome itself, that is also a very big FASTA file. So it's very hard to find something in that FASTA file. So the same applies here. These are really big files and it's very hard to find something in these files. So this is why you have to process them. So first of all, what we're going to do with SUM and BUM files is we're going to sort them. So the order of the reads in the SUM and the BUM file at this point is the same order as in the FASTQ file. So the reads are ordered according to the ID, according to the location of the cluster on the flow cell. So still that. Now, for the downstream tools, it's much more convenient to put the reads in a different order, to put the reads in the order that they appear on the genome. So first the reads that map to chromosome 1, then the reads that map to chromosome 2, and so on. You can easily see that for visualization, this will be much easier for the tool than in a random order. And the same thing is true for the counting. So that's what sorting will do. It will put the reads in the BUM file in this order. So it's very big files, these SUM and these BUM files. So just like we did with the FASTA file that contains the reference genome, here also we're going to index these SUM and BUM files to make them easier to access. That is also a step. And then for some applications, not always, but for some applications, you're also going to flag the duplicates. What does this mean? It doesn't mean that you're actually going to remove duplicates from the SUM or the BUM file, but you're going to flag them so that the downstream tools know, oh, I already processed this read. And that will make the processing for the downstream tools much faster and more accurate. It will make your counts, because that's typically what you're going to do after the mapping. You're going to count. It will make your counts more accurate. But I will come back on that, on these flag duplicates. So, but these are typically the 3 steps that you do to process your BUM and your SUM files. So there are many tools that allow you to do so. The most used tools for doing this are called SUM tools and Picard. I will come back on this in the next slide. You will also check the quality of the mapping. Just like you checked the quality of the raw reads, the quality of the sequencing basically, you're also going to check the quality of the mapping. And the tool that is used the most for doing that is called Qualimap. And it's very similar to FastQC. And then the nicest part of the whole workflow, you're finally going to visualize something. So you're going to visualize the results of the mapping. And for that, we're going to use a tool called IGV. So but we will start with the processing, the sorting, the indexing, and the flagging of duplicates. So when you sort and index your BUM and your SUM files, what is generated? Well, still a BUM or a SUM file, typically a BUM file in this case because they are more compact. But the order of the reads in the BUM file has changed. So it's a sorted BUM file, and when you index your BUM file, a separate file is generated, and that separate file is called a BY file. And BY stands for BOM index. So the index will be in a separate file with the same name as the BOM file, but instead of having a name .bom, it will have a name .by. Okay, so here again you see the 3 steps that you do to process your SUM and your BOM files. So I will come back here on this one, the mark duplicates. I told you for most applications you do this, but not always. So for DNA-seq data, you're going to mark your duplicates. For RNA-seq data, it's not necessary to do this. Because for RNA-seq data, the duplicates in most cases will have a biological origin. So you want to keep them in, and you do want to count them. Why do you need to do it? Well, because if the duplicates are not biological duplicates, but technical duplicates, they will distort your counts. So, and for DNA-seq data, you're also going to calculate counts. You're going to count how many times a variant occurs in your reads. That's a count. Well, that count has to be based on biological duplicates and not on technical duplicates, because then it will be not accurate. So this is why you do this step. Okay. So the 2 tools for doing these kind of manipulations on your BOM and your SUM files are called PICAR and SUM tools. So it's not one tool, they're basically both a set of tools, and they will have tools for sorting and indexing BOM and SUM files. So for PICAR, this is called sort SUM. In SUM tools, this tool is called sort. You will have tools for just doing the indexing on a file, a BOM file that is already sorted. So some mappers generate an unsorted BOM file or SUM file, and then you have to sort and index it yourself. Some mappers will do the sorting already for you if you ask them. And then you just have to do the indexing on that BOM file. And PICAR also has a tool for marking duplicates. How to make a distinction between technical duplicates and biological duplicates? Well, you can't. In some cases you can, but it's not possible to make a distinction between duplicates that arise from PCR amplification and real biological duplicates. They will both be duplicates, they will have the same sequence, so yeah, there you are. So it's not feasible to do it, and this is why they don't mark duplicates for RNA-seq data. So in RNA-seq data, you will have many biological duplicates, but of course there also will be technical duplicates. There's not really a way to avoid that. But because you cannot make the distinction, people say, oh, we're going to play it safe and we're not going to remove them. We're going to assume that they're all biological. That's the best thing that you can do here. Now for DNA-seq reads, you don't expect to see duplicates. So there, if you see them, well, they have to be of technical origin. So there people do remove them because you don't expect to see biological duplicates. So when you see duplicates, they're probably technical duplicates. So that's the distinction. The only type of technical duplicates that you can identify are the optical ones. So what are optical duplicates? Optical duplicates are duplicates that arise from clusters that are on the border of multiple tiles. So if a cluster is on the border of 2 tiles, well, it will be in 2 pictures and it will be analyzed twice. But there you can recognize them because they come from clusters that are very close to each other. And then you can see, this is a technical replicate. But for technical replicates that arise from PCR, you have no way to recognize them. For RNA-seq data, you can use these UMIs. So you add one UMI, one unique UMI for every transcript. So if the transcripts are duplicated during the library prep, they will have the same UMI. And this is a way to make a distinction in RNA-seq data between technical replicates and biological duplicates. But in bulk RNA-seq data, these UMIs are not heavily used. So in single-cell RNA-seq data, they are always used. In bulk RNA-seq, not that much. But you can use them, and it's a big benefit because it will allow you to make a distinction between the biological and the technical replicates. So of course, if you have them, then you can remove your technical replicates from your RNA-seq data, and then you would have to mark duplicates there also.